Welcome back, guys. Welcome to another video sessions from kame biology. In this video lecture we will talk about the life cycle of the polio viruses, right? So now let me write life cycle of polio viruses. Now first of all, what is polio viruses? Polio viruses are those viruses which can cause paralysis inside the human or inside babies, or we can say at the age of 1 year babies, or at the age of 1 year children. At that age it can cause the paralysis. Fine. Now how the life cycle of the polioviruses or occur? Now let me explain. First of all, polioviruses are actually, they are non-enveloped viruses. They have no envelope. They have just capsid, and within the capsid, protein capsid, they have the RNA, and they carry single-stranded RNA. Fine. So now let me draw the structure of the polioviruses, that how the polioviruses are actually. Fine. Now this is the poliovirus. This is the structure of the poliovirus. This layer, or this layer, outer layer, as I draw, these are the capsid, and inside the capsid they just carry what? They just carry the positive mRNA, or positive single strand RNA. Fine. Now this virus is first of all, how this viruses enter inside the healthy host cell. First of all, the infected person will carry these viruses. Fine. Now once the healthy host, or once the healthy person will contact with the infected, this poliovirus infected person, so this viruses will easily be transmitted, you can say through water contamination or through fecal contamination. Fine. So once these viruses will enter to the healthy person, so they will be enter to the mouth, and from the mouth then they will be reached to the digestive tract, and from the digestive tract then they will reach to the intestine. Fine. So now in the intestine, these polioviruses will start their life cycle, because in the intestine there are nutrients, or there are some other availabilities are present, through which or there are some requirements are present for the poliovirus, through which the polioviruses will start their life cycle. Fine. So that's how the polioviruses will reach to the intestine, and here they will start their life cycle. So how? If I draw the structure of intestine, so now let's suppose intestine structure will be just look like in this form. Sorry, these are the intestinal cell. Fine. So now these are the intestinal cell. What actually happen? The virus will attach to the receptor. There are some receptors which are present on the intestinal cells, and once they attach to the receptors, so they will be. Now there is one other question. As I told you that naked viruses will be entered through cell-mediated endocytosis. If you remember, go back to my channel and watch that how the naked viruses are actually enter inside the host cell. They are entered through the cell-mediated endocytosis. What does it mean? It means that they will be engulfed, and once they engulfed, so there will be enveloped inside the there will be enveloped outside the naked viruses, and only the RNA will be released. Fine. Means the endosomes will form. But in polioviruses, but in this case, in case of polioviruses, what happened? They just attach. So now let me draw the structure again. They will just attach. Fine. So they will just attach, and what happened? They will just attach. They will release their RNA. Now their RNA are released. The poliovirus are not engulfed, or not engulfed through cell-mediated endocytosis. They will just attach and release their positive mRNA. Fine. So now this positive mRNA is used as a template, and this positive mRNA are actually produced. What this positive mRNA are produced? These proteins. These are the proteins. Fine. So now these are the protein. Different proteins they can produce. Within each proteins, there are RNA dependent polymerase also. Fine. So now within these proteins there are RNA dependent polymerase also. RNA dependent polymerase. Fine. So there are RNA dependent polymerase protein also present, or we can say enzyme are also present inside these proteins. So what happened? These RNA dependent polymerase enzyme will sit on this positive mRNA, and they produce what? They produce negative strand. Fine. They produce negative strand, and now this negative strand is used as a template. We need this negative strand, and this negative strand is now used as a template, and then transcription process will occur, and once the transcription process will occur, then they produce what? They produced again more and more. So we get more and more copies of what? We get more and more copies of positive single stranded RNA, because we need that. And now these positive mRNA then release structural protein. These are what? These are the structural protein. Let me write structural protein. So once we get the structural proteins, and after that these mRNA and the structural protein will combine together, and what we get? Then we get what? Then we get the poliovirus. So now polioviruses are formed, and within this capsid there are the single stranded mRNA, or we can say positive mRNA or single stranded RNA. Now once the polioviruses are formed, a lot of polioviruses once formed, so they will put pressure on the host cell. Means the host cell in this case are the intestinal cells. So they will put pressure on the intestinal cell, and the intestinal cell will burst, and all polioviruses are released. Once all polioviruses are released, so they will be now entered to the bloodstream. Let's suppose this red one, these are the blood vessels. Fine. So these viruses are enter inside the blood vessels. So a lot of polioviruses are enter inside the blood vessels, or we can say they are enter inside in the bloodstream. Fine. So once these polioviruses are enter in the bloodstream, so they will be moves to the nervous system, or we can say then these bloods are carried by this by these streams, or we can say that once these polioviruses are enter inside the blood vessel, so they will be carried to the nervous system or to the brain. So now let me write moves to brain and cause nervous system destruction. Nervous system destruction. Destruction. So then the nervous system will be disturbed, or the nervous system will be disrupted. Once the nervous system are destroyed by these polioviruses, then what happen? Then disease will happen. Means paralysis disease will happen. Fine. So that's how the polioviruses can cause the paralysis. Or these are the life cycle of the polioviruses. Brain are made up of neuron, means neuron cells. So brain are made up of neurons, so they will disturb the neurons. Means so once the neurons are disturbed, so finally paralysis will occur. Fine. So that's all about the life cycle of the polio viruses. Now let me explain all of the cycle with in Urdu or in Hindi language that what happened. असल में आपके पास ये पोलियो वायरस जो है ये आपके पास नॉन इन्वेलप वायरसेस है. फाइन, नॉन इन्वेलप वायरसेस है. पहली बात ये, इसमें आपके पास सिर्फ और सिर्फ आउटर मेम्ब्रेन मतलब ये कैप्सिड होता है और इसके अंदर आपके पास पॉजिटिव मैसेंजर आरएनए होता है. ये वायरस जो है अटैच हो जाता है इस इंटेस्टाइन सेल्स पर. सबसे पहले ये वायरस जो है माउथ के ज़रिए एंटर हो जाते हैं, फिर डाइजेस्टिव ट्रैक्ट में सफ़र करता है और फिर इसी तरह ये जो है इंटेस्टाइनल जो सेल्स होते हैं वहाँ पर ये अटैच हो जाते हैं. जिससे ये अटैच हो जाते हैं ये सिर्फ अपना पॉजिटिव mRNA रिलीज़ करता है. जैसे कि हमने डिस्कस किया था कि नेगेटिव वायरस जो है वो एनगल्फ हो जाते हैं और फिर एंडोसोम सा बन जाता है, लेकिन इस केस में जस्ट ये यहाँ पर अटैच हो जाता है और फिर ये सिर्फ और सिर्फ पॉजिटिव mRNA प्रोड्यूस करता है. जब ये पॉजिटिव mRNA प्रोड्यूस करते हैं तो चूंकि इस इंटेस्टाइनल सेल्स में अपने पॉलीमरेज एंजाइम मौजूद होता है तो ये पॉलीमरेज एंजाइम इस mRNA पर यानी सेट हो जाते हैं और हमारे पास ट्रांसलेशन का प्रोसेस आकर हो जाते हैं. जब ट्रांसलेशन का प्रोसेस होता है तो हमारे पास बहुत से प्रोटीन्स बन जाते हैं. इन प्रोटीन्स में हमारे पास आरएनए डिपेंडेंट पॉलीमरेज प्रोटीन्स या एंजाइम होते हैं. ये एंजाइम क्या करते हैं, इस आरएनए पर सिट हो जाते हैं और इस आरएनए पर ये आरएनए एज अ टेम्पलेट को यानी ये आरएनए एज अ टेम्पलेट इस्तेमाल होता है. फिर इसी तरह हम क्या हासिल कर लेते हैं, हम हासिल कर लेते हैं नेगेटिव mRNA. अब ये नेगेटिव mRNA हमारे पास एज अ टेम्पलेट इस्तेमाल होता है और ये जब एज अ टेम्पलेट इस्तेमाल होता है हम बहुत सारे पॉजिटिव mRNA हासिल कर लेते हैं. जब हम बहुत सारे पॉजिटिव mRNA हासिल कर लेते हैं तो फिर ये पॉजिटिव mRNA जो है फिर मज़ीद ट्रांसलेशन का प्रोसेस करके हमारे पास क्या बना लेता है, स्ट्रक्चरल प्रोटीन. तो हमारे पास स्ट्रक्चरल प्रोटीन बन जाते हैं. ये स्ट्रक्चरल प्रोटीन और ये mRNA हमें चाहिए और जब ये दोनों मिल जाते हैं तो हमारे पास मैच्योर क्या बन जाता है, हमारे पास मैच्योर पोलियो वायरस बन जाते हैं. ये पोलियो वायरस क्या करते हैं, असल में ये पोलियो वायरस यानी जो होस्ट सेल होता है यानी ये वाला जो इंटेस्टाइनल सेल होता है उस पर प्रेशर डालते हैं. जब जैसे ही प्रेशर डाल देते हैं तो ये जो वायरस है ये यानी सेल को बर्स्ट कर लेता है और ये वायरसेस रिलीज आउट हो जाते हैं. जब ये वायरस रिलीज हो जाते हैं तो ये वायरसेस ब्लड वेसल में चले जाते हैं. ब्लड वेसल में ये ब्लड स्ट्रीम में चले जाते हैं. ब्लड स्ट्रीम में ये सफ़र करते रहते हैं और ब्रेन में चले जाते हैं. ब्रेन में ये न्यूरॉन्स को डिस्टर्ब कर लेते हैं यानी नर्वस सिस्टम ये पूरा डिस्टर्ब हो जाता है. जब नर्वस सिस्टम डिस्टर्ब हो जाता है तो हमारे पास क्या हो जाता है, फिर पैरालिसिस डिजीज़ होती है. सही. तो ये था आपके पास लाइफ साइकिल ऑफ द पोलियो वायरसेस. आई होप दिस वीडियो हेल्पफुल. इफ़ यू हैव क्वेश्चन सो राइट इट इनटू अ कमेंट. आई विल ट्राई माय बेस्ट टू गिव यू आंसर. एंड इफ़ यू लाइक द वीडियो प्लीज सेट द like button, share my video and subscribe my channel to get more interesting video lecture like that. Thank you.